A malignancy aggressively engineers its own localized hostile environment. It builds dense networks of chaotic blood vessels and actively secretes acidic fluids to create a fortress that ensures its survival. To build this fortress, the tumor exploits specific biological loopholes. It hijacks normal cellular metabolism to secure endless energy, evades the body's immune surveillance, and constructs an acidic extracellular matrix that protects it from attack. Many clinical misconceptions regarding integrative oncology equate the field with passive wellness supplements, a mild regimen of vitamins meant to generally support health. In practice, integrative oncology utilizes highly concentrated, multi-targeted biochemical agents to systematically dismantle the specific oncogenic pathways that keep a tumor alive. This is a first principles attack. By understanding exactly how the cancer defends itself, we can deploy specific botanical and nutritional compounds to neutralize each protective layer. To execute this, we will analyze the mechanisms of 6 distinct interventions: berberine and high-volume juicing, modified citrus pectin, agaricus blazei, EGCG, and flaxseed. Mapping these 6 agents demonstrates how their distinct mechanisms overlap to physically and chemically strip a tumor of its protective microenvironment. The first target for disruption is the cancer cell's power grid, specifically its mitochondrial and glucose receptor infrastructure. Berberine, a highly potent botanical alkaloid, functions as a direct, multi-targeted metabolic restrictor against this grid. Once introduced, berberine aggressively targets the cell's energy sensing systems, locking onto and forcefully activating the AMPK pathway. This activation triggers an immediate downstream consequence. The stimulated AMPK node sends a suppression signal that severely inhibits the mTOR and PI3K-AKT signaling cascades. With its primary energy supply severed, the cancer cell experiences rapid mitochondrial depolarization. The structures degrade as the power is cut. At the molecular level, this specific action replicates the glucose-lowering and tumor-suppressing actions of the pharmaceutical drug metformin. By severely restricting the energy supply and downregulating key growth proteins, berberine systematically starves the cell, forcing the malignancy to undergo programmed cell death. Beyond the cell's interior, malignant proliferation relies heavily on its surrounding extracellular matrix, a highly acidic, oxygen-deprived terrain. To physically alter this environment, integrative protocols utilize intensive, high-volume juicing. This acts as a mechanical delivery system for high-yield phytochemicals. Mechanically breaking down plant cell walls and separating out insoluble fiber serves a strict clinical purpose. It allows up to 90% of the therapeutic nutrients to be absorbed directly into the bloodstream within 15 minutes, completely bypassing the energetic burden of digestion. Wheatgrass juice is a critical component of this process. Its high chlorophyll content possesses a chemical structure nearly identical to hemoglobin, allowing it to rapidly oxygenate tissues and reverse the anaerobic conditions in which cancer thrives. This oxygenation works in tandem with epigallocatechin-3 gallate, or EGCG, the most abundant bioactive polyphenol derived from green tea. EGCG launches a secondary metabolic attack by actively inhibiting the enzyme glutamate dehydrogenase. This stops the tumor from utilizing the amino acid glutamine as an alternative energy source. Combining rapid extracellular oxygenation with glutamine restriction physically alters the biological terrain. The tumor is stripped of its protective acidic shield and effectively cut off from its secondary fuel lines. As a tumor grows, its greatest threat comes from its ability to migrate and spread throughout the body. This spreading relies on galectin-3, a specific protein that facilitates cancer cell clumping, tissue migration, and the formation of new blood vessels. Modified citrus pectin, or MCP, is a complex carbohydrate derived from citrus peels, chemically altered into smaller molecules to allow for proper intestinal absorption. MCP's primary anti-neoplastic mechanism is structural. It actively binds to and inactivates the galectin-3 receptors protruding from the cancer cell surface. By capping these receptors, MCP physically prevents malignant cells from adhering to one another and stops them from latching on to the inner walls of blood vessels. Clinically, administering 15 grams of MCP daily has demonstrated significant success in managing disease progression, notably slowing the doubling time of prostate-specific antigen levels in prostate malignancies. Acting as a non-toxic structural barricade, MCP restricts the tumor's ability to metastasize, keeping the patient's immune system from being overwhelmed by an expanding cancer load. With metastasis restricted, the focus shifts to the patient's own innate immune defenses, which the malignancy has forced into a dormant state. The medicinal mushroom Agaricus blazei is utilized here. Its active agents, complex carbohydrates like proteoglycans and beta-D-glucans, do not act as direct toxins to the body. Instead, these polysaccharides function as biological response modifiers. They systemically stimulate and amplify the patient's natural killer cells and macrophages to trigger a selective tumoricidal effect. But immune cells can only attack what they can recognize. Deep within its nucleus, the cancer cell uses abnormal genetic silencing to hide its vulnerabilities and protect itself from apoptosis. EGCG acts as a powerful epigenetic modulator to reverse this very mechanism. EGCG aggressively inhibits specific silencing enzymes, known as DNMTs and HDACs, physically uncoiling and exposing the suppressed DNA. This inhibition effectively turns the body's natural tumor suppressor genes back on, rendering the malignant cell completely vulnerable to cell death enzymes, known as caspases. By stimulating natural killer cells and uncoiling suppressed genes, these agents convert cold tumors, those that are invisible to the immune system, into hot targets that are recognizable and destroyable. The final vector of attack focuses on disrupting the hormonal signaling pathways that drive hormone-sensitive malignancies. When flaxseed is ingested, intestinal microbiota metabolize its plant lignans into mammalian lignans, specifically enterolactone. Enterolactone acts as a selective estrogen receptor modulator. It competitively binds to estrogen and androgen receptors, favorably altering hormone metabolism to slow tumor growth. However, integrating flaxseed into a clinical protocol reveals a critical debate over bioavailability, whether to use extracted flax oil or freshly ground whole seed. Cancer cells frequently lack delta-5 and delta-6 desaturase, the specific enzymes required to efficiently convert the alpha-linolenic acid found in flax into an anti-inflammatory state. Because of this blocked pathway, the extracted oil can remain trapped in a pro-inflammatory omega-6 state, a condition that is particularly dangerous during radiation therapies. To safely bypass this metabolic limitation, clinical trials show patients must utilize 25 to 50 grams of freshly ground whole flaxseed daily to achieve tumor suppression and reduce metastatic spread. We have observed a precise systemic dismantling, energy lines severed by berberine, metastasis barricaded by MCP, immune recognition restored by agaricus and EGCG, the matrix oxygenated by juicing, and hormonal pathways blocked by whole flaxseed. These compounds act as powerful sensitizers, weakening the tumor's defenses to make it more vulnerable to conventional oncology treatments. For example, EGCG safely eliminates cancer stem cells, the hidden and resilient cells responsible for treatment resistance and disease relapse. Weakening the cell's metabolic and structural architecture makes it more vulnerable to attack. Berberine and modified citrus pectin have both been shown to synergistically enhance the tumor-killing effects of doxorubicin by disabling the protective proteins that would otherwise shield the cancer cell from the drug. They achieve this heightened efficacy while simultaneously fortifying the patient's healthy biological terrain, buffering the body against systemic toxicity. Today, clinical protocols heavily utilize this synergy during aggressive standard treatments, including intense radiation therapies. Integrative oncology applies her rigorous, targeted science. It systematically strips away the metabolic foundations of a malignancy, ensuring the host's innate defenses have the exact tools they need to prevail.